Hey everyone, and welcome back to the final episode of season
My
7,
gosh.
Twisted. I can't believe we are at to the end of season 7. I feel like we say this all the time though, and we say this all the time that we say this all the time, but we are so on. We're always like, oh my god, it's so crazy. It's like the last
We're
episode,
at
but
the end.
I'm actually really sad this season is coming to an end because I've really enjoyed it. I think I was someone who absolutely hated ethics in uni, like the ethics module.
Yeah.
Yeah, that shit was so boring. But I kind of feel bad now that, like, honestly, now I've learned about all of these stories, I'm like, I have a
Yeah, you're
newfound
like,
love
thank
for
God.
ethics
Yeah.
and realize how important it is. So I'm excited to get into today's final story because I feel like we've saved a really, really important topic for the last
Mm-hmm.
episode. And it is a topic that you guys have brought up to us a few times. So I'm very excited to like learn more about it. But before we get into today's episode, I have a question for you guys and also for Taz because I actually wrote this question down in my notes to ask Taz back on Christmas Day.
Oh.
Like, I'm sat at the dinner, like the Christmas dinner table. I'm literally waiting for my Christmas dinner, and my sister's fiance and my brother's girlfriend, they started having a conversation, and it just got me thinking about something. And the question is, why are there no old nurses? Like every single nurse is like in their 20s, 30s. And I just got into some really deep thoughts about the fact that you never see old nurses. And I'm like, is it just me?
This is such an interesting question. Also so random, what the hell. I've actually got a really crazy question that I asked myself once, and like no one's ever really been able to give me an answer. So we'll get to that in a
Okay.
minute. Well,
Okay.
answer your question first. Am I like being, I don't know, like I guess being a nurse is really hard. That's my only guess.
Do you think, wait, maybe it's because it's like physically a lot, right? Maybe that's why?
Yeah, they're running
Okay.
around. It's mentally like tolling as well.
So do you think like there's a dropout of nurses, like as in like you
Yeah,
only do this job for like a certain period of time and then you're like, I can't. I mean, that would make sense because
sure.
I feel like that's like being a nurse must be like one of the hardest jobs, like literally.
Yeah, the oldest nurse I've known gave me a vaccine, and like so that's not mentally tolling. And also, she was still only like what 50. It's
And
not
was it
old
in a
old.
GP surgery?
Yeah,
See, I'm talking
so
about like
really
like
old. Isn't it?
nurses in hospitals. Like they're never. The reason I thought of this question is because my brother's girlfriend said about how her granddad and her grandma actually met because they were both nurses, and she said about how her granddad was actually a nurse until he was like 55, and I was like, oh my god, and he worked in a hospital, and I was like, I never see old nurses in hospitals. But I think you're
To
probably
be
right.
fair
I
though, do
reckon.
we ever really? I never really go to a hospital.
I mean, fair, same. But I feel
So we
like
could just be
okay.
like talking out of our arse right now, just being like, "There's no old nurses," and like everyone who's like a nurse and a doctor is like, "Yes, there
Yeah, there
is."
is. Oh, guys, okay. If you're someone who goes to a hospital regularly or like you work in a hospital or something like that, can you tell us like are there older nurses? And if not, do you guys have any theories as to why that might be? Is it because it's like one of those careers where it has like a high dropout period after a certain amount of years? Or is it that the job is like physically demanding and yeah, they move to like GP surgeries or they move and do other things? Can you guys let us
Mm.
know? Taz, what
Maybe
was
they
your
stop
question?
being front facing because they're like they've done the work on the ground, you know. And now they're in charge of the nurses, so they just boss
So it's
around
a
the nurses,
natural
but like
career
the
progression,
patients don't see
maybe.
them. Maybe.
Yeah, no, that's a
Maybe.
good
Let us
point.
know, guys, if you know anything about nursing.
Yeah.
Let us know. My question is completely different.
Okay.
But I had this thought one day and I was just like, yeah, why is that not a thing? My question is, why do we not have any savory drinks? They're all sweet. Every drink that we have that's marketed is sweet. We don't have a single savory drink.
So- wait. To counter that though, would you not say that like English breakfast tea is savory until you add sugar?
No.
No? If you don't have any sugar,
This is
that's
a wild point because you could say the thing about coffee.
but then what does savory mean? Like
Like black coffee, that's quite
yeah.
savory with no sugar.
What does savory mean?
Well, I
I mean,
meant
this is
like, I don't know, like bacon or something.
Bacon flavored drink.
Like bacon Coke or something.
Nah, this is actually such a valid point, and I've never really thought about it. But I would say that English breakfast tea is, if savoury drinks exist, I think English breakfast tea without sugar would be one.
Yeah, I actually never really thought about that. My mind was more going towards like
Cold drinks.
I don't know, like fizzy drinks.
Yeah.
Like you don't have, there's no like savoury fizzy drink.
No, that's true. Why don't we have a bacon fizzy drink?
Yeah, or like I'm trying to think of another savoury.
No, that's actually such a valid point, because if we like it in food,
imagine crisps. Imagine crisp flavours, but in sparkly drink form.
Salt and vinegar.
You like a crisp, so why
salt
wouldn't
and vinegar,
you like,
soda water,
like sour cream and onion?
And no, that's such a good point. Can I have a fizzy salt and vinegar, please?
Yeah. It's basically the same thing, isn't it? Just less calorific, maybe.
I think that's a really interesting philosophical question that you bring up on this podcast. And this is why we do this podcast, because you ask the important philosophical questions of our time. Do you know what I mean? Like, wow. You're so insightful, Taz.
I think I have a notes page of like questions like that.
I want to be in on this notes page. This has got me thinking, I wonder how many of our listeners have additional philosophical questions. So,
oh my God,
sorry if you've already dropped a comment about the nurses, or maybe you haven't, and here's another opportunity to drop a comment on this episode. Can you let us know, like, what is your philosophical question that you don't know the answer to? Because we could discuss them on the podcast. So let us know, what are your questions? Do you guys have a notes
also
page?
Let us know what savoury drink you would drink.
Yeah, I would defo try the salt and vinegar fizzy.
Yes, salt and vinegar. I think like anything vinegary would actually be good. Like I know someone who drinks vinegar out of like the pickled gherkin jar. She drinks
a few people do that.
Someone make a pickled gherkin fizzy drink. I'm intrigued.
So am I. I'm
what
gonna be
a fun
about
way
this
to
all
start
day.
the episode. I really want to know everyone's thoughts now.
Same.
These are the types of scientific questions you're asking on this podcast, guys.
Well, this is what I'm saying. We only ask important questions on this podcast. Like, you just don't need any other podcasts but this one. But now for real. Okay, we're chatting rubbish. We're chatting rubbish. Let's actually get into some proper science. Let's get into the topic of the final episode of season 7. Let's get into it.
Yeah.
Welcome to the So Cultured podcast,
where we explore science and tech unfiltered.
We're your hosts, Liv and Taz, two girls who love STEM and spilling the tea.
Each season, we take on a new theme, whether it's busting conspiracy theories, celebrating women in STEM, or breaking down the science in your favourite sci-fi films.
And of course, in every episode, we spill the laboraTEA,
where we share your wildest stories from the world of science.
Let's get into this week's episode of So Cultured.
Right, we're ending the season on maybe a bit of a lighter, it kind of has a bit of a positive spin on it, but like everything, it kind of originates with some very questionable ethics, but it does kind of have like a more positive side of the story and positive end to the story, but we'll get into it. But a lot of you might have heard of this topic before, so if you have, let us know, and let us know your thoughts on it as well, because there are a lot of conversations around it and you'll figure out what those are when I get into it. So let's get into the episode. So if you've ever taken a vaccine or benefited from cancer treatment, or even learned basic biology, there is a strong chance you've been helped by a woman you might not ever have heard of. And her name was Henrietta Lacks. So she was 31 years old. She lived in the United States in the era of segregation, and she unfortunately died in 1951. But parts of her are still alive today, and that's not metaphorically. That is literally.
I'm so intrigued. I've heard about Henrietta Lacks, but I feel almost like ashamed to say this. I'm not going to lie. But if there's any other listeners who feel the same, like, we're here together. But I don't think I've ever actually learned in depth about this story. And as we said last week's episode, we get so many comments asking us to discuss this. So I was so excited when you said that it was the topic of today's episode. So yeah, let's get into it. Who exactly was Henrietta Lacks, and how did she have such an impact on science? That was a very broad question, but we can go from the beginning.
Yeah, so let's start at the beginning. So if you are a scientist or you worked in lab work, you may have worked with a cell line called the HeLa cells. And the HeLa cells are what we scientists call an immortalized cell lines, used in scientific research continuously since the early 1950s. And they are the oldest human cell lines still in use, and they are among the most widely used biological materials in the history of science. So under the right conditions, so nutrients, warmth, oxygen, HeLa cells do not stop dividing. That is what it means to be an immortalized cell line. And while most human cells divide, there's a limited number of times they can divide before they start aging and dying. Whereas HeLa cells, they just keep going day after day, year after year, literally a decade after decade. And their durability, rapid growth, and ability to survive indefinitely under laboratory conditions has basically made them foundational to the advances across medicine, genetics, virology, cancer research, and biotechnology. There are now infinitely more HeLa cells in laboratories around the world than there were cells in Henrietta Lacks' entire body when she was alive.
Gosh.
But although the cell line has contributed to scientific discoveries that have benefited millions of people worldwide, its origin is inseparable from broader historical issues surrounding medical ethics, informed consent, racial inequality, and the ownership of biological materials, which is basically why we're talking about this in this season of Twisted.
Oh my god. Yeah, okay. So I know like a little bit about her story, but for like any of the listeners, they probably were listening to like the first paragraph of, like, if they don't know anything about this story, they're probably listening to like the first paragraph thinking like, am I listening to the wrong series, because this sounds great, like what is the issue, do you know what I mean? But obviously we're going to get into it, and it's such an interesting story just from like the kind of like base knowledge that I have. So yeah, so can you talk about how this cell line seemingly had such a great impact?
So to understand why HeLa cells changed everything, you've actually got to understand what science couldn't do before 1951. So for decades, researchers have been trying to grow human cells outside the body. They could isolate them, they could place them in petri dishes, but within days, sometimes hours, the cells would die. So whilst animal cells were more readily available, obviously, there was not an indefinite supply of human cells, because that was pretty much impossible, right? Because obviously the cells, they kept dying after a certain number of divisions, and like there's just not an indefinite supply of human cells that can like logically be applied to science, I guess. So this was a massive problem when doing science research. If you couldn't keep cells alive, you couldn't test drugs safely, you couldn't study viruses in a controlled way, and you couldn't observe how cancer behaved at a cellular level. And this limitation placed really strict constraints on experimental design. So researchers could not reliably study long-term cellular processes, they couldn't observe disease progression over time, or conduct repeated testing on identical genetic material. So drug development, toxicology studies, and cancer research were largely dependent on animal models, which obviously don't accurately always reflect human physiology either. And the inability to sustain human cells in vitro, so like in a petri dish, represented a fundamental barrier to progress in biomedical science. And scientists were looking for something that for years seemed impossible: a human cell line that could survive indefinitely in the lab.
Oh my gosh, I never even really appreciated that in general. Do you know what I mean? I've never even really like given much thought to having a human cell line that can survive indefinitely. That's really interesting. So can we talk about how these cells actually came about? I mean, obviously you've mentioned Henrietta Lacks. These are called the HeLa cells. Well, I call them HeLa. I think either is fine. I mean, but I'm assuming obviously there is a link between Henrietta Lacks and HeLa cells, obviously. So can we talk about how these cells actually came about?
Yeah, 100%. And I think that's a really important part of the story. Like, where did these cells actually come from? Who was Henrietta Lacks? And that's kind of what we want to get across in today's episode. So Henrietta Lacks was born in 1920 in Clover, Virginia, and she grew up relatively poor, working tobacco fields in a family shaped by generations of racial and economic inequality and segregation. So by 1951, she was actually living in Baltimore with her husband and children. And unfortunately, she began experiencing irregular vaginal bleeding, so she went to Johns Hopkins Hospital. And at the time, Johns Hopkins was the only major hospital in the area that treated Black patients, though the care was still delivered in segregated wards. Now, medical practice during this period did not require physicians to obtain informed consent for the collection or use of tissue samples removed during diagnostic or therapeutic procedures. So Henrietta was unfortunately diagnosed with cervical cancer, and this was later identified as an aggressive adenocarcinoma. But during her treatment, a physician named Lawrence Wharton Jr. removed small samples of tissue from her cervix, both healthy and cancerous. Now, Henrietta, which is important to note, was never told that these samples were taken. She never gave consent, but at the time, this was fairly standard practice. Now, those samples were sent down the hall to a scientist named George Otto Guy, head of the tissue culture laboratory.
That is crazy. Like, because obviously informed consent is such a big thing now. Like, it's just so insane to think that someone can just take part of your cervix and not even ask you, let alone tell you what they're going to be doing with that. That's crazy. So what actually happened then? Like, why did they want to take, like, did they purposely want to take these cells for something in particular? Like, what did he do when he had them?
So I think, like, at the time, a lot of this stuff was very opportunistic. So I think it was fairly standard practice. If people were having procedures where tissue were removed, then they were just kind of like experimented on in the lab, and the patient didn't need to give consent for this. Obviously, now we know it's not like that at all. And this is kind of like foundational in this story as well. So George Guy had actually been trying for years to grow human cells in the lab. He was a cell biologist who had spent decades attempting to grow human cells in culture, and his laboratory employed a then novel technique involving roller tubes, which allowed cells to be bathed continuously in nutrient-rich medium while rotating to improve oxygenation. So when he received Henrietta's cells, like everyone else, he expected them to die after a certain number of divisions. But instead, something extraordinary happened. Henrietta's cancer cells divided, and then they divided again and again and again, and every 20 to 24 hours, the number of cells doubled. So one Petri dish became 2, 2 became 4, 4 became 8, and they didn't slow down, they didn't degrade, and they didn't stop. For reasons that were not fully understood at the time, these cells behaved unlike any human cells scientists have ever seen. Rather than dying after a limited number of divisions, the cells continued to proliferate and showed no signs of senescence. So this marked the first creation of a human cell line capable of indefinite replication in vitro, so in a Petri dish. And George Guy realized he was looking at something unprecedented. He called his colleagues and he said, "I think I have the first immortal human cell line." Now, the cells were designated HeLa following a standard laboratory convention that used abbreviated donor identifiers to kind of maintain anonymity. For many years, the identity of the donor remained unknown to the public, and the cell line was actually mistakenly attributed to fictitious individuals like Helen Lane or Helen Larson.
That's insane. You know, when you said like the cell line's immortal, what does it actually mean for a cell to be immortal?
So HeLa cells are considered immortal because they bypass the normal cellular mechanisms that limit division in what we call somatic cells, so like not cells that are immortal. So basically in healthy human tissue, cell division is constrained by telomere shortening, DNA damage checkpoints, and programmed cell death pathways. And that's kind of like a self-protection element. Over time, like cells and the DNA within them get damaged. So basically it kind of prevents DNA damage and ultimately cancer. And cancer is basically where it goes wrong. So cancer cells, including HeLa cells, often acquire mutations that essentially disable those controls. So in the case of the HeLa cells, several genetic abnormalities contribute to their behavior, including chromosomal instability, aneuploidy, and altered regulation of telomerase activity. These features allow the cells to replicate indefinitely under appropriate laboratory conditions, and over time, HeLa cells have actually accumulated additional mutations, leading to the emergence of multiple substrains. But despite this genetic divergence, all HeLa cells in existence today are descended from the original tumour cells taken from Henrietta Lacks in 1951.
Wow, see, that's so interesting. I did not know that there had been substrains that have emerged from her cells. Like, I did not know that. Obviously, as I said, like, I've heard about her story, but I didn't know that. That's so interesting. But that only kind of signifies to me that these must have been a cell line that was widely adopted within the scientific community, and so many people were using them to get to the point where it's acquiring so many mutations that you have multiple substrains. So was that the case, that it was like really quickly adopted by the scientific community?
Yeah, it really was. So obviously recognizing the significance of the discovery, George Guy distributed HeLa cells freely to researchers all around the world. And at the time, there was no established framework for intellectual property rights related to cell lines. And Guy believed that widespread access would accelerate scientific progress, and he was not wrong. But he did not patent them, he did not sell them, he literally gave them away. Any scientist who asked essentially received the HeLa cells along with instructions on how to grow them. And so within years, they were literally everywhere. They spread across the United States, Europe, across the world. And HeLa cells were rapidly adopted due to their robustness and ease of culture. And laboratories had previously struggled to maintain human cells. They now had access to a reliable, reproducible, experimental system. So it was like truly revolutionary. So this widespread distribution marked a real turning point in biomedical research, effectively standardizing human cell culture as a routine experimental tool that it is today, basically. But unfortunately, Henrietta Lacks meanwhile, she was dying.
That's so sad. And like, it's a really odd one, because like, I don't know how to feel about him. So he didn't patent it, he wasn't charging people for it. So it's almost like he wasn't directly exploiting her and her cells for his own gain. Like, he was maybe doing something. I mean, I guess we'll never really know what was going on in his head, but I guess from the way the story's being told, it seems like he just genuinely wanted to help people, and maybe he was also victim of a system that just had no proper ethics in place. Do you know what I mean? But it's so sad that ultimately I'm guessing she unfortunately passed away from her cancer, because it sounds like it was a really aggressive cancer.
Yeah, absolutely. So Henrietta's cancer was aggressive, it was relentless, and so despite treatment, it spread rapidly throughout her body. And unfortunately, she died on October the 4th, 1951, just months after her diagnosis at just 31 years old. And I think that is like the crux of this entire situation, where like we know now today that HeLa cells have done so much and they've helped advance science so much, but it's come at a cost, and the cost is someone who unfortunately passed away from cancer, but also had no idea where these cells were going, and like obviously the ethical considerations with that. So that is like the main crux of this whole episode and this whole topic. So she never knew that her cells were still alive despite passing away, obviously. She never knew that they were being used in laboratories across the globe. And in fact, her family wasn't even told. And for decades, they would actually have no idea.
Do you know what? I take back what I said a minute ago, because I almost feel like, as if his intentions were really true, as far as he's concerned, it was probably a great legacy that he thought he was helping her leave behind. So why would he have no interest in telling the family? Yeah, these cells could have done great. They could have cured cancer. That's great. But ultimately, who knows if that's what she would have ever had wanted. Do you know what I mean? Like, we're all just assuming, oh, that's a great thing, and it's done all of these amazing things. It's helped advance science, but ultimately she might not have ever had wanted that for herself. And that's the whole point, right? And it's also interesting because I wonder if she was picked particularly as well. Do you know what I mean?
This is why it's such a big topic, and this is why I do think what you said earlier about him kind of being part of a system where the ethics just aren't there, and informed consent, especially in that time when there was like segregation. So I just think like the whole topic is just like there's just so many facets to it,
which is so like incomprehensible to us now. Like,
yeah,
so
exactly.
Yeah, we just can't even get our heads around that.
No, I agree. And I honestly think like there's so many ways that you could look at this. But I do agree, like if this was such a revolutionary discovery, then like surely the family would want to know. And like we will go on to learn their thoughts and feelings about all of this once they do find out later on in the episode. But yeah, I think obviously the ethics just weren't there. They've developed so far from like these awful situations that we've covered in this season. I think that's why this is such a big conversation.
Yeah, no, I 100% agree with you. And ultimately, I think, you know, it's such a tragic ending that she not only lost her life, but lost her life so young. Like 31 is so young. I mean, that's like close to our age now. So it's such a sad story. But I don't know, I guess, can we talk a little bit about the impact of her cells? So let's talk a little bit about what actually came from a result of the work that used her cells.
Obviously, they were tragic and essentially twisted circumstances, but actually a lot of good came from the use of her cells. So I'm just gonna talk about some of like the more major contributions, because they're like literally used to this day all around the world. So new discoveries are being made like all the time using her cells, but I'll delve into like the most kind of prominent ones. So one of the earliest and most consequential applications of the HeLa cells was in the development of the polio vaccine. So in the early 1950s, polio was one of the most feared diseases on earth. It literally paralyzed children and it killed thousands. So Jonas Salk was working on a vaccine, but testing its efficacy and safety required enormous numbers of human cells that could be reliably infected by the virus. So HeLa cells turned out to be perfect for the job. They were easy to grow, easy to infect, and when infected with polio, they died in a way that made results easy to measure, or what we call in science a clear, measurable cytopathic effect. So many cells were needed that actually a large-scale production facility was established at Tuskegee University, one of the first places capable of mass producing human cells to supply laboratories nationwide. Less than a year later, Salk's vaccine was ready for human trials in 1954. So this was just 3 years after Henrietta's passing.
Wow.
The vaccine was declared safe and then effective shortly thereafter, and polio would eventually be pushed to the brink of global eradication, and HeLa cells made that all entirely possible. Now, following their success in polio research, HeLa cells became a central tool in virology. They have been used to study a wide range of viruses, including HIV, measles, mumps, herpes simplex virus, Zika virus, and human papillomavirus. In HIV research specifically, HeLa cells were genetically modified to express the CD4 receptor, which enabled the scientists to study viral entry and replication mechanisms, and these experiments contributed to a deeper understanding of HIV pathogenesis and inform the development of antiretroviral therapies. Now, obviously, the most obvious applications were their use in cancer research. So as a cancer-derived cell line, HeLa cells have played a significant role. Researchers have employed HeLa cells to evaluate the efficacy of chemotherapeutic drugs, radiation therapy, photodynamic therapy, and targeted molecular treatments for cancer. And they've also been used to identify cancer-associated markers in our RNA to develop more kind of RNA screening systems. And because of their aggressive growth characteristics, HeLa cells have served as a model system for understanding tumor biology. Now, in 1965, HeLa cells were used in pioneering experiments that fused human cancer cells with mouse cells, creating the first human-animal hybrid cells. And these experiments allowed scientists to associate specific genes with individual chromosomes, and HeLa cells were also the first human cells ever cloned. So this work laid the critical groundwork for things like gene mapping, cloning, and long-term cell storage and handling. So this ultimately contributed to the development of really large-scale breakthroughs, including the Human Genome Project, cloning Dolly the sheep, and most importantly, in my opinion, IVF as well.
Wow, I did not know that specifically. I didn't know about Human Genome Project, Dolly the sheep, or IVF.
Yeah, and actually one of the most wild ones that I researched about HeLa cells was they were actually used in the early years of space exploration. So they were sent aboard satellite missions to study the effects of microgravity on human cells.
So many amazing things have obviously come from these cells. And I almost feel bittersweet about it, because obviously it's incredible and so many amazing discoveries. But also it's you don't know how to feel about it, because you don't know how she would have ever felt about it. And I guess obviously that's the main crux of this story, right? Is it's such an amazing thing, but it's almost it doesn't feel like something you can celebrate, because you don't know if she would have celebrated it. Because obviously she never got to see, obviously, the impact that her cells had on people's lives. But I guess related to that, obviously earlier you mentioned the family. So I feel like this is a good time to ask, how did because obviously he never told them. So how did the family eventually find out, and how did they feel about this situation?
By the 1970s, obviously, as we've kind of established, HeLa cells had become so widespread that actually another problem emerged. They were almost too good at surviving, and due to their rapid growth and resilience, HeLa cells were found to contaminate other cell cultures, often without other researchers realizing it. So entire experiments, researchers were unknowingly studying HeLa cells instead of what they thought they were studying. So studies revealed that many cell lines believed to originate from different tissues or donors were in fact HeLa cells, and this discovery prompted widespread reevaluation of experimental results and led to ultimately improved laboratory practice and authentication protocols. But obviously, we need to find a way of identifying these contaminated cell lines. So Johns Hopkins, they actually contacted the Lacks family to request DNA samples, and the family didn't understand why, and they weren't given clear explanations. But in the process, they learned for the first time that parts of Henrietta Lacks were still alive, and those were the HeLa cells, which I just think is wild. This revelation was, as they have described, confusing, frightening, and deeply upsetting that they didn't know about any of this.
Very confusing. I was going to use the word confusing, extremely confusing. I cannot even imagine finding that out. It would just be such an unsettling, odd experience. But I'm assuming this was fairly recent, as in ethics would have progressed somewhat between them needing to request the DNA samples. And so I think it's even crazier that they're bringing them up basically being we need some DNA samples. And then even still, the family still feel confused. They feel like they're still not getting clear explanations. It's just even in whatever that time frame was, that still wasn't good enough.
Yeah, no, this is the impact that Henrietta Lacks and her cells had. Even by 1971, which is obviously when this cross-contamination
So like 20
event
years later.
was revealed. Yeah, 20 years later, like medical ethics hadn't really moved on, and actually it was like the family finding out and the impact of that kind of changed the course of medical ethics.
Wow. How brave of them as well, because yeah, I mean it's very clear that John Hopkins like are trying to be intentionally vague about it. So how brave of them to like speak up, and obviously they were such a huge part of this change in medical and scientific ethics as well, like the family themselves as well as Henrietta.
Yeah, and unfortunately as well, Henrietta Lacks' story was not unique, but it was obviously one of the most visible examples of how like 20th century medical research was happening. So tissue samples were actually routinely collected and used without patient knowledge, particularly among marginalized populations, and informed consent as we understand it today did not exist. So patients were rarely told when their tissues were used for research. Poor patients and patients of colour were disproportionately affected. And ultimately, the hospitals benefited, the research benefited, and companies did profit off of these cells by selling them, but the family and Henrietta's family specifically did not. So as HeLa cells generated kind of enormous scientific and commercial value, the Lacks family continued to struggle with even just basic access to healthcare.
That's so insane. Yeah, when you lay it out like that, it's like, that's insane to hear. Like, it actually makes you angry. Like, it literally makes you angry.
Yeah, they weren't a wealthy, privileged family. They were like, yeah, struggling for the very basic things. And it's just so crazy that this legacy that Henrietta left was not benefiting the family at all.
Right. And I mean, ultimately, that's probably why she was targeted, right?
Well, exactly, yeah. And like I said, these kind of practices did disproportionately affect marginalized communities.
Exactly.
So yeah, there is definitely strong themes of racism throughout this, as we've covered on this season, like quite extensively, I feel. So as HeLa cells became commercially valuable, ethical questions started to emerge regarding ownership, compensation, and importantly, patient rights. So these issues were later addressed in legal cases such as the Moore versus Regents of the University of California, which actually ruled that individuals do not retain property rights over discarded tissues.
Interesting. But her tissues weren't discarded. And also, it's like, does that still stand when there was no informed consent given?
Yeah, so at this point, obviously, informed consent still was not an established thing. It was just established that discarded patients' tissues were not like the ownership of the patient. But yeah, like you say, Henrietta Lacks's tissues weren't discarded tissues, but and they were also taken without her knowledge. But basically, her case became part of a broader reckoning and obvious cause for concern among people. And I do get it in that patients now who consent to have their tissues used for research couldn't then be like, give me the money, if it paves way for things like very profitable drugs or like treatments for things. But obviously we're talking about informed consent, and that didn't happen for Henrietta. So like obviously today you'll have a patient and like it's kind of similar to organ donation, right? You'll have a patient who is happy to donate their body to science or donate their tissues for science. But when they do that, it's kind of like a charitable donation. It's not like I'm investing this part of my body to reap the rewards in the future if you like create a cure based off my tissue. Like that's just not how it's done. It's kind of voluntary donation, but obviously it's informed consent, which obviously wasn't a thing for Henrietta. But in 1991, the common rule was established in the United States, and that established standards for informed consent and human subject protection in research. And in the 2010s, following the publication of Henrietta Lacks' genome, concerns about genetic privacy prompted the creation of the HeLa Genome Data Access Committee, which includes members of the Lacks family, alongside scientists and the National Institutes of Health representatives. And this committee reviews applications for access to Henrietta's genomic data, basically. So for the first time, actually, research subjects, or their families in this case, had a seat at the table and involvement in the research that was going on.
See, I think it's great, obviously, that they've involved the family, but honestly, it feels like a bit of an afterthought to me. I personally still feel like that is crazy to publish, like to sequence and publish her genome.
It is just exploitative because there's
Yeah.
no consent, and it's like, oh, you know, this is
Yeah.
ours now, and it's like, well, no, it was never yours to begin with, you know, to do anything
No,
with.
I
That's the
agree.
problem.
I agree. My question is, are her cells still used today? Because I feel like they are. I feel like I've heard.
Yeah, they are still a
Yeah.
very widely used cell line. By the early 21st century, actually, more than 60,000 scientific publications had referenced research
Wow.
involving the HeLa cells, with like hundreds added each year, basically. And their contributions extend across nearly every major area of biomedical science, as I've kind of already covered. And HeLa cells, they still continue to be used today, and they actually were included in early stage research during the COVID-19 pandemic, reinforcing their ongoing relevance. And actually, one of the most significant discoveries made using the HeLa cells came decades later. So for years, scientists believed that cervical cancer was caused by the 𝗾𝘁𝗁𝗃 virus type 2. However, after testing cervical cancer tumor samples and not finding any traces of this virus, scientist 𝗾𝘗𝗁𝗃, a German virologist, began to think that maybe human papillomavirus, or HPV, was the cause instead. Actually, in 1983, he proved his theory by identifying HPV-16 and HPV-18 DNA, two types of HPV that now are commonly associated with HPV-related cancers, in HeLa cells and other cell lines originating from cervical cancer tumors and original tumors as well. And it actually earned a Nobel Prize, and it led to the development of the HPV vaccines, which I think they're actually offered to boys now, but obviously if you're our age and you're female, you might have received this vaccine when you're about like 11, 12, before you become sexually active, to prevent cervical cancer. And the research on the HeLa cells were crucial in developing this vaccine because it identified key viral components that were essential for an effective immune response. So this actually led to the creation of vaccines targeting high-risk HPV types, and it significantly reduces the mortality of HPV-related cancer by as much as 70%. So in a tragic irony, the cancer-causing virus that ultimately killed Henrietta helped save literally millions of others. And it could have been you or me, Liv. Obviously, we have had the HPV vaccine, and so like we've been protected against, hopefully, cervical cancer. So yeah, it's just crazy to think that that's like another legacy that these cells and essentially Henrietta have left.
Yeah, no, 100%. Get your smear test, ladies, as well. Like, I feel like some of our listeners need their reminder because as you were talking, I was thinking I need to go back for my checkup. So here's
Do you know what I realised when we were doing this, when I was doing this research, is, you know, it's called like a pap smear. I'm like, it's because it's the human papilloma virus they're looking for. Pap.
Wait, I've never thought about that.
I never put that two and two.
Oh my god! Do you know what? It's things like that I just think I'm actually dumb. Like, how do I not make these connections in life? Like, I'm actually stupid.
I didn't even realise till I was doing this research. I was like, oh yeah, obviously, because they look for HPV, don't they?
Interesting. How have I never made that connection? Get your pap
smears,
girls.
Get your pap smears.
Get your pap smears.
Cancer screening.
Very important.
Not hard.
But yeah, I think, I mean, you said it. Tragic irony. That is how I feel about it. I mean, obviously, it is great that her cells were able to do so much for science. I mean, you've given countless examples in today's episode, but I can't help but think that economically these kind of like big pharma companies, they must have profited like massively from the use of her cells. And I guess that's another thing that's giving me like this really uneasy feeling about this. It's like that thing of, okay, this is amazing, but it's almost like you just don't want to celebrate it in the way that you feel like you should. Do you know what I mean?
Yeah, 100%. I think a lot of people when they learned about Henrietta Lacks' story and also the family and how they found out. They were like, wow, like you've been given no opportunity to have any involvement with these cells or the development of the research, and they didn't know about it for such a long time. And also, I think critically, ultimately, we're talking about a family who didn't have even access to basic healthcare due to their
that actually angers me so much. That
economical
makes me so
means.
angry.
And then you have pharma companies that are basically profited. But actually, in more recent years, and I'm talking like in the past 5 years, the Lacks family actually sought accountability from the companies that profited from the HeLa cells without consent. So in 2021, the estate of Henrietta Lacks filed a lawsuit against 𝗾𝘂𝗁𝘂𝗁 Scientific in Baltimore Federal Court, alleging that the company unjustly enriched itself by selling HeLa cells which were taken without consent in 1951. Now, the suit asserted that 𝗾𝘂𝗁𝘂𝗁 Scientific knowingly profited from the cells. Now, 𝗾𝘂𝗁𝘂𝗁 argued that this was outside the statute of limitations, but Lacks' lawyers' argument was that the cells were still being used and profits were still being made to this day. And the settlement was reached on the 1st of August 2023, which actually would have been Henrietta Lacks' 103rd birthday. But the terms of the settlements, aka how much the Lacks family would have got paid, are confidential, but both parties stated that they were pleased to resolve the matter outside of the court. And actually, following this settlement, the family's legal team indicated that they would now pursue lawsuits against other companies that have profited from the HeLa cells. So I think, like, get it, girl. I just hope they're getting paid now. That's all I'm going to say.
100%.
Anything positive that's going to come from this for the family, obviously knowing that the cells have and Henrietta have such a insane legacy, but I think let's try and do something right here and pay the family. Not that you can put any monetary value on this kind of thing, but like they need some monetary gain from this, I think.
Yeah, and even if it's not about the money, it's like accountability for these companies who I can't even begin to imagine the amounts of money these companies made. Like, I cannot even begin to imagine. So accountability, 100% necessary. And I think it's so amazing and admirable that the family pursued that.
Yeah, 100%.
And I hope they continue to. Like, I really hope they continue to. Obviously, we know about the HeLa cell line because, like, it helped advance science in so many, like, disciplines. It helped with so many amazing things. But also, I like to think that another reason we know about Henrietta Lacks' story and we know about the HeLa cells is also because of her family. So I think that's great as well. And that's like
actually there
worth
was like
mentioning.
a really important book that was published. Basically, there was this reporter, I can't remember her name, I think her last name was 𝗾𝘂𝗁𝗂𝗁, and it was actually off the back of the 𝗃𝘃𝗁𝗃 syphilis study.
Wow.
And how that like gained public scrutiny. And actually this journalist ended up making a book about Henrietta Lacks. She worked with Henrietta Lacks' daughter to basically uncover the story of the HeLa cells, to kind of establish what happened to Henrietta Lacks, and also like how the cells came about and how they were distributed so widely. So if you want to learn more about that, I'll put the link to the book that the journalist published in the link below. And I think, that will probably go into a lot more detail. But we'll round off here. And we know Henrietta Lacks obviously was not a scientist, and she did not choose to contribute to research. And unfortunately, she did not live to see the impact of what was taken from her. But her cells have saved lives across generations. There are people alive today because of HeLa cells. They represent one of the most significant tools ever developed in biomedical research, and their ability to survive indefinitely transformed how scientists study disease, test treatments, and understand human biology. And at the same time, their history underscores the importance, again, of ethical standards, transparency, and the respect for the individuals whose bodies have contributed to scientific progress. And understanding HeLa cells requires acknowledging both their extraordinary scientific value, but also in the context in which they were obtained. And that is a balance that continues to shape modern biomedical research.
Such a lovely summary. Yeah, I feel like this is such a complex, bittersweet, nuanced story, but it's a story that I'm so glad we got to feature on this series and hear more about. So yeah, thank you so much for that, Taz. And for now, we are ending this season with our final Ask Us Anything, and we have some crazy things to answer to. So let's get into it.
But I'm worth it, 'cause I'll slip into your dreams tonight. So give me your
Okay, let's get into our last Ask Us Anything for season A. I actually have 5 questions here, Taz, to get through because all of them are funny. And I'm also so intrigued as to what the listeners are going to think of these questions and what some of the listeners' answers would be to these questions. So guys, don't forget that you can leave a comment on Spotify or you can DM us on any of our socials with your answers to these questions because we want to hear your guys' thoughts on these as well. But let's get straight into it. So Taz, the first one is, would you rather redo your entire PhD or explain your research to a drunk stranger every weekend for a year? That is a hard
I don't know about
one.
you, but this one's actually kind of easy for me.
Why?
Oh, you just said this is a hard one. Because I feel like I do that anyway. Like, I do that every weekend. Like, not being funny, every time I'm in an Uber, the Uber driver be like, what do you do? And I'll be like, I do science
You need to start lying. I just lie. I just make something up. Like, I don't want to explain my science to you. I make something up. Like, I'm debating redoing the entire PhD. Like, are you joking? I don't want to have to explain it to you every single weekend because they always think it's great. And I'm like, I'm so done with it. Like, obviously, there's times when I love it. But if you catch me on a week where I'm fed up with work and I've got to explain it to you and you'll do
your entire PhD again. Although, to be fair, we've had this conversation before where we're like, oh, I reckon if I did my PhD now, I'd get it done in like 2 months.
Quick time. I'd be done in 3 months. Like, what are you talking about? We're in the days of AI as well. Like, are you joking me? We did our PhD with no AI. Like, obviously, I'm glad.
ChatGPT.
We had no Jenny. We had no Jenny. Imagine, like, we had no Jenny to help us get through. Like, it's just, yeah, I can't even. So I genuinely think I could get it done like that. But would I want to? The thing is, if I'm explaining my research to a drunk stranger every weekend, am I drunk every weekend? Do I get an Aperol out of it? You don't have to be drunk
every weekend.
But I get an Aperol out of it.
You get an Aperol out of it, yeah.
Oh, I'm going to explain my research.
This is the thing. I'm basically out every weekend being like, yeah, I do this, I do that, I do
Taz, we need to come up with a cover story for you. I think you should say that you are just come up with like a really obscure, like random, say you work in the circus. Like, genuinely.
No one's gonna believe that, are they?
Why not?
Yeah, true. I feel like a lot of listeners will probably relate to this. But during the pandemic, anytime you said that you're a scientist, they were like, oh, so what do you think?
Oh, Taz, I can't even imagine.
I feel like it's the same bloody thing, you know? And you're just like, just don't ask me. I actually, in the pandemic, I did make up my job because I was like, especially the Uber drivers, they were like, do you believe in the vaccine? It's dangerous. And I was like, oh, God, obviously I do. The amount of conversations where I had to be dragged into that. Never.
No,
again.
I literally can't even imagine. I cannot even imagine. But in all seriousness, I reckon I would choose to explain my research to a drunk stranger every weekend for a year. I think I'd have to go with that.
Yeah.
Guys, let us know in the comments, what are you doing? Because although if you've done your PhD now, you probably could do it quick time. What would you rather? I don't know. Okay, moving on to the next question. This one says, if your thesis was a movie genre, what would it be?
Ooh, oh well, actually this is easy for me. Mine would be like zombie apocalypse because it's God, yes. Rabies, like outbreak, pandemic preparedness, vaccines, you know? So mine would be like World War Z or something.
The movie plot could be my cat brings a bat into, sorry, OG listeners will get this reference if you don't understand. But the movie plot needs to be my cat brings a bat into my house and then the bat bites
me. The pandemic starts.
For the non-OG listeners, right? This was back when we wait, this was before we actually started the podcast. This is before we started the podcast. One day, right, I was sat in my bedroom and my cat, I'm not even joking you, brought in a bat. She brought in a bat. So we were hearing all this commotion in the hallway. Me and my sister, we both open our bedroom doors and there's a freaking bat flying around in the hallway upstairs. And we were like, oh my God, this cat has brought in the bat. Anyway, I said to Taz, I was like, oh my God. And she was like, be careful that it doesn't bite you. Then I convinced myself that this bat was going to bite me and it was going to give me rabies. And then Taz, being Taz, was telling me about how rabies would kill me. And like, if I get bit or scratched, then I need to go to hospital. Then it was just like this whole spiral. Taz was spiraling, I was spiraling. So that's going to have to be the plot.
But not only that, I told you to, if the bat died or something, I told you to send it to me and I would test it.
Yeah, she asked me to put the bat in Tupperware and send it to her. Imagine you're the postman.
I think we both spiraled in that moment, but
yeah, we did.
You know, that is how the movie would start. It would start like that, and we would both be like scared. You would be the first zombie, like rabid zombie, and then it would be like World War Z kind of vibes, and then I would be in the lab trying to like create a cure. Like, what's his name? Was it Brad Pitt in that movie? I can't remember, in World War Z.
Don't be asking me, girl.
Creating a vaccine. That'll be us. You'll be a zombie.
That would be
I'll
us.
be a scientist.
Mine would be, obviously the genre is going to be horror. Like, my PhD was literally a fucking horror movie. But it's one of those horror movies where I keep coming really close to death, but I'm not quite dying. Do you know what I mean? Like, I get stuck in
it's like Groundhog Day, where you're like stuck in the same day every day.
Yeah,
and it never ends.
trying to fix the same experiment over and over again. And every day you wake up and do the same shit and expect different results. That is literally it. But no, mine would be one of those horror movies where I keep getting put in like horrific situations and like it's a really long drawn out, and then it finally gets to the point where I'm like, just take me, I'm done, like I'm gonna die, I've accepted this fate, and then something saves me and I'm almost like, for sake, like I actually have to go through this again. And
you're that person in the horror film who goes and checks out the scary sound and then ultimately dies.
No, I survive. I survived until the end.
Yeah,
I'm like, I'm like that character that like just shouldn't have survived, but I did somehow.
The final destination.
That's
character.
Like the PhD graduation. That you get to your graduation and you're like, how the f am I here? That is literally, that would be me.
Yeah. I love how I did mine like related to my research and you just did yours on your PhD experience. So I love that.
Guys, let us know what you pick. What movie genre would represent your thesis or your PhD? Let us know. Okay, next question. What is the most delusional thing you believed early on in your PhD? Mine is that it would open up job opportunities.
God, so true. And
that I'd be paid a lot of money once I graduated. Like,
a lot of money. That is such a good one. Like, I'm like, surely if I've got a PhD on my CV, I'm going to be getting P's. No.
No, I'm here. Like, I've run out of coffee and I need to buy some new Nespresso pods, but I'm in my overdraft. I'm not even joking. I'm actually not even joking. It's tragic. I think it's on top of everything, I think it's the job opportunities for me. Like, I was really convinced, like, I'm going to have a PhD. Like, what are you talking about? Like, I am the 1% in the population. I am great. And then it's like, actually, I think I'm dumber than when I started. Like, I genuinely think my intelligence
my biggest
regressed.
Misconception is that if you have a PhD, you're smart.
I think you get dumber. I genuinely think you get dumber. Like, I'm being serious. Like, you just get so focused on one thing.
So specific that you're just like, what? I don't know anything else about. Like, this podcast has taught me more than my bloody PhD taught me. So
yeah, no, literally, I agree. Like, I've 100% learned more on this podcast, and I hope some of our listeners can also relate. But yeah, I mean, there's so many delusions when you start a PhD. No offense to any of our first years or people who want to pursue a PhD. There are so many great things about doing a PhD, but with it comes many delusions.
Yeah, and I honestly think hindsight is so funny because I look at like first year PhD student me and I'm like, lol, you thought you were the shit, but you were just like a meager first year PhD student. Do you know what I mean? Like, obviously it's an achievement getting there, but like, I feel like you go through so much in a PhD that when you come out the other side, you're like,
the pick me energy is gone. Pick me
yeah,
baby,
the
she's
pick me
she's
energy
well
is
and
gone.
truly
You're
gone.
just, yeah, you're just
Okay.
downtrodden and beaten up. Sorry, we're making.
No, it's just
PhDs
funny.
are actually so good, guys. Don't let this put you off.
No, we're only joking. It's just funny to laugh. Like, we just enjoy it. Yeah.
It's just our dark humor. It's fine.
Related to this, the next question is, what behavior in academia would be unacceptable in literally any other workplace? I actually have an answer to this. It just comes straight to my brain, which is, why the fuck do lecturers dress the way they dress? Because I don't think that would be acceptable in many other workplaces, because I swear I had one lecturer who, I don't think he ever washed his clothes. They were always dirty. They had several holes in them. They were obviously from the 1980s. Like, it was an Adidas hoodie from the 1980s, but not the cool vintage kind, which you go to like a vintage shop and you pay like £100 for. It's like, it had holes in, it was dirty. So my thing is, why does everyone in academia dress badly and unprofessionally? Like, nobody dresses nice. My supervisor, he dresses nicely, and I think he's one of the few people who actually, I go into work and I'm like, yeah, he looks put together, he looks clean, he looks like he's a professor.
Yeah, I agree with you that that is something that would not fly outside the academic setting, but low-key, I do love that, because
I mean, I do, don't get me
turn
wrong,
up
girlie.
to work
Like this.
looking absolutely ratchet and like no makeup, hair is just greasy and slicked back like this, and
I think you look
I
presentable.
don't know. Also, I think obviously when I was like working in high containment facilities, I was like showering out and like I would look like oh, that's not that's another one. I used to walk around. It relates to like what you look like around work. I used to walk around work with a towel on my head.
Oh my god, what? Because you just
Like drying.
come out of the lab and you had to shower?
Yeah.
You need to give the listeners more context, because they're probably like, why is she...
Well, so obviously, basically, I used to work with viruses. So I'd work in like high containment facilities, and that would mean like when you're in there, you're in like a boiler suit. You've changed out your clothes, and then obviously when you come out, you leave the boiler suit on the dirty side of the containment facility, and you shower, and then you get back dressed into your normal clothes. But obviously, showering involves you have to wash your hair, you have to like wash everything with soap. And not only did I have like ridiculously dry skin, because the air in there is like just negative pressure or something, so it's always taking the moisture out of the room, but also showering in that water was like so drying. And so yeah, I had like crispy dry skin, and also I had a towel on my head for the majority of the time that I was there, because I'm not walking around with like sopping wet hair. I mean, they did have hair dryers, but honestly, I would like go in the morning and then have lunch, and then I would go in the afternoon. So like, what's the point?
I agree.
So I just wear a towel on my head.
Wear that towel, girl.
And sometimes I would wear the towel
and
home, because I'd be working in the lab really late. And then I'd shower out and I'd be like, I just want to go home. So I'd put the towel on my head and drive home with the towel on my head.
I can imagine you just walking through the car park with the towel on your head and people being like, what the hell?
Yeah.
This kind of stuff, it does fly at unis though. It flies at unis. All right, last question, which I don't actually know that me and Taz are going to be able to answer on air, but it says, if you could send one anonymous message to your supervisor, what would it say?
Oh my gosh.
I don't think I can answer that.
If you're listening, I zipped my lips and threw the key away.
I can I could comment on a previous supervisor, but recent supervisors, I would tell him he's great. He's absolutely great. I could say some things to previous supervisors, but maybe me and Taz shouldn't do that on our podcast that has thousands and thousands of listens. But you guys can definitely let
It's
us
not
know.
so anonymous as well if we just announce it on a podcast.
Exactly. So yeah, me and Taz probably shouldn't be answering that question on this podcast, but you guys can definitely answer that question by dropping a comment on our Spotify or just DMing us on Instagram or TikTok at soculturedpodcast. Or if you fancy it, you could leave them a little message in our anonymous laboraTEA link, and we will read them out on the podcast next season. So with that rounds off episode 10 of season 7. So Twisted is officially done,
It's done.
which means we are now going to be taking a short break before we return for season 8. We are open to requests. We are still refining our idea for season 8. So if you guys have any particular topics or themes that you would really want us to cover, then definitely let us know. And we also will be doing a brand new batch of laboraTEA stories for season 8. So now is your chance to get your story in if you want us to react to and listen to it on season 8. You can do that by clicking the anonymous link in today's episode or video description. And obviously, if you guys have been enjoying season 7 or any of our other seasons so far, then here's your reminder to make sure that you are following us on whatever podcast platform you listen to us on so that you don't miss out on season 8. And also make sure you're following us on Instagram and TikTok at soculturedpodcast, because we will be announcing the theme for season 8 there, and you can also stay up to date with us so that you know when we're coming back for season 8 by following us on our social media platforms.
But thanks for joining us for season 7, and we shall see you guys soon. Bye.
Bye everyone.